Well, I think I would like to give you more my perspective, a global perspective of what I've seen over 5 years, because this series started as a CAR-T meeting, and then TCRs got tacked on with a hyphen. And that was a very small group of scientists, a very small area of activity when it started. And now one sees that the two approaches for adoptive cell therapy are equal parity. I mean, the TCRs have really taken their place and bring real highlights, as well as the starting clinical data also to the forefront at the CAR-TCR meeting. The TCR people have the advantage that they can learn a lot from the CAR-T field because they're marketed products, and one is getting real life data from what's happening with the therapies in patients that are no longer recruited into clinical trials, but are being treated in different countries and different clinical settings. So all of the challenges around the logistics and marketing and interacting with many different centers and regulatory authorities in different countries, that really helps, I think, the TCR field to streamline its understanding of the problems, and that can help ease things. We started a first TCR-T trial in Germany. Germany, unfortunately, hadn't really participated in the CAR-T trials for the marketing authorization of the two main products, and therefore the infrastructure wasn't established. Having a center set up that it can do an apheresis, the logistics to get that to a production center, the tracing, the tracking, the identity, and all of those things. And so it was very helpful to be able to have examples from the CAR-T. So I think that's a very, very positive fusion. And of course, what's being learned from the marketing side will also flow into making things more efficient for the TCR arena. And of course, the forefront that pushes forward with the TCR arena, the real tailwind, is the possibility to apply these to solid cancer indications, where it doesn't look like that's going to be very easy with CAR-Ts, just because of the limitations in the number of target antigens. And then, of course, the real scientific highlights that start to come out of this CAR-TCR platform is how one can modify the cells to give them more power, and what can one do in conjunction in combination therapies to achieve overcoming the immunosuppression present in a tumor microenvironment. So those would be the main highlights that I would say, just this tremendous progression and the feedback from CAR to TCRs.